Hi, my name is Melissa, and today we're going to be talking about a paper called Synthetic Lethality of TNK2 Inhibition in PTPN11 Mutant Leukemia. This is a study done by Jenkins et al. Okay, so this study sought to investigate interactions between a non-receptor protein tyrosine phosphatase called PTPN11 and a non-receptor kinase associated with cell division control, TNK2. So TNK2 is thought to be an oncogene when it becomes overexpressed, which is associated with poor outcomes for patients. TNK2 is also thought to be involved in regulating downstream pathways that involve SRC and EGFR, which is an epidermal growth factor receptor. PTPN11 is known to be involved in the pathogenesis of juvenile myelomonocytic leukemia, which will henceforth be referred to as JMML. This is a leukemia that begins in blood-forming cells in bone marrow. PTPN11 is also involved in acute myeloid leukemia, which is going to be called AML. PTPN11 has a PTP protein tyrosine phosphatase domain and two SH2 domains, which include TYR542 and tyrosine 580 residues. So PTPN11 normally undergoes autoinhibition. So this is where SH2 N-terminal blocks the catalytic PTP domain. And usually PTPN11 is only activated by phosphorylation of C-terminal tyrosine residues. And this leads to the activation of downstream pathways like MAP kinase. However, mutated PTPN11 does not experience autoinhibition. And this results in increased phosphatase activity and proliferation of mutated cells by increasing downstream signaling for cell survival and division. And this contributes to pathogenesis of JMML. 85% of JMML cases involve mPTPN11 and other genes associated and like it that are related to MAPK. So PTPN11 mutations are also present in about 5 to 10% of AML cases. This study sought to also understand the signaling upstream of PTPN11 in JMML and AML cells. Furthering treatment research for these illnesses is important because currently it's limited and the treatment options are very limited. JMML comes back in 50% of transplant recipients within only 2 years, and AML treated with chemotherapy only has a 24% chance of a 5-year survival. So new therapies are needed. New research has suggested that mutated PTPN11 may be sensitive to dasatinib. And previous studies have shown its promising effects of reducing mouse lung cancer by reducing the interaction between PTPN11 and GAB1, which is a signal transducing protein. So researchers wanted to know if it could inhibit upstream proteins of PTPN11. Another study suggested dasatinib could inhibit RAS signaling and GM-CSF, which is granulocyte macrophage colony stimulating factor. And they wanted to know if it could be inhibited in JMML cells, possibly due to dasatinib's inhibition of SRC family kinases. So this investigation served to further explore a mechanism that could be used by dasatinib, a multi-kinase inhibitor, to treat mutated leukemia cells. Okay. So after they confirmed that that mutation was there, they were able to assay ex vivo against panels of 66 small molecule kinase inhibitors, or siRNAs, for 3 days. Okay, so now we're going to take a look at the results of these tests for this first set of results. So for the first test, they found there was significantly reduced cell survival in the presence of siRNA that were targeting TNK2. So we can see in figure 1b, this shows the viability of individual kinases. And it indicates that JMML cells depend on TNK2. So treatment of this patient's cultured cells with small molecule inhibitors showed a sensitivity to dasatinib, and we can see this in figure C. And it had a mean inhibitory concentration, as you can see on the side there, it says IC50. That's mean inhibitory concentration. And it was 36 nanomolars compared to the median of 401 for a diverse sample of 151 leukemia patient samples. So these cells were basically nearly 10 times more sensitive to dasatinib. So for the second test, they found significantly increased MAPK phosphorylation in cells containing either PTPN11 mutants compared to the wild-type version. And you can see that in figure D. So if you find where they have the two mutants, so the G60R and the E76K, you can see that if we go down to where there's a blot for the phospho P44 to 42 MAP kinase, you can see it's a lot darker in those spots, which means there's a lot more phosphorylation and activation of that pathway. And the result for the last test in this section, this assay showed in figure 1E that GM-CSF-induced colony formation in cells with either mutation indicated a gain-of-function mutation, which basically just meant that they were experiencing way more cell proliferation if they had the mutated versions of PTPN11 compared to the wild type, which we see on the left is a lot less colonies. So we're going to talk about the next part of their experiment. So So for the first test, there was co-expression of mutant PTPN11 and TNK2, and it led to more phosphorylated P44 and 42 MAP kinase than cells expressing the mutant PTPN11 or TNK2 alone. It also showed increased phosphorylation of PTPN11 at residues tier 542 and 580, and we can see this in figures 2A and B. And it reduced the phosphorylation of TNK2 at tier 284, which was involved in activating TNK2 when co-expressed with PTPN11. But this was more pronounced with the mutant PTPN11, and we also see this in figure 2C. So let's look at figure A closely for a second. So if you can find at the top of this figure, you can see where they're being co-expressed and where it's co-expression with the wild type. And there we don't see as much phosphorylation of MAP kinase compared to when we have our mutant PTPN11 with the TNHK, or TNK2, sorry. And there we see a lot more phosphorylation of MAP kinase. And then over here in figure B, that's where we see, again, the same thing. We see when it's combined, they're co-expressed, and there's a mutant PTPN11, then we get a lot more activity of the phosphorylation of the MAP kinase pathway. And then we also see how what is it called? We see that there's reduced phosphorylation of TNK2 at tier 284, and we see that here when they are co-expressed. All right, so test 2 results. So active TNK2 is required to enhance mutated PTPN11 signaling, and as active, mutant TNK2 led to phosphorylation of P44 and 42 MAP kinase, and inactive TNK2 stopped this. And then the results of test 3. they found there was an increase in phosphorylated P44-42 MAP kinase and decreased TNK2 phosphorylation where there was co-expression of the mutant PTPN11, specifically the E76K, and TNK2. But when either tyrosine domain was mutated, there was less MAP kinase phosphorylation. It decreased to baseline levels. So mutant PTPN11 requires both the TIR542 and 580 residues for downstream signaling. And again, in figure D, we just see how these two molecules could co-precipitate with each other. Okay, the next section we're going to talk about is inhibition of TNK2 reducing signaling through PTPN11, RAS, MAP, and kinase. So the first test they did was to find the impact of TNK2 inhibition on mutant PTPN11 signaling. The method they used for this one is they co-expressed mutant PTPN11 or wild type with wild type or gatekeeper mutant TNK2. So the gatekeeper mutant is resistant to dasatinib and AIM-100, which is another inhibitor. They treated the cells with dasatinib and AIM-100 and did an immunoblot. We're going to see that coming up in the next one. So the next test they did is they looked at inhibition of PTPN11, or they asked if inhibition of PTPN11 blocked the dephosphorylation of TNK2. And the method they did for this is they used SHP099 which is an allosteric inhibitor of PTPN11. The third test they did for this section is they thought because mutant PTPN11 favors active conformation, it may even more so in the context of TNK2 co-expression. So the way they looked at this is they used wild-type PTPN11, which can be phosphorylated by SHP099, but it's stuck in its inactive form, and so it cannot signal the MAP kinase pathway. So let's look at the results for this one. So the next set of results we're going to look at is TNK2 as a functional target for PTPN11 mediated transformation. So the first test they looked at was to determine if TNK2 can enhance transformation potential of mutant PTPN11 and whether targeting TNK2 can stop colony formation due to PTPN11. The way they looked at this is they took bone marrow mouse cells and these were transduced with mutant PTPN11 with or without TNK2. And then for the second test, they tested if PTPN11 mediated colony formation could be stopped with TNK2 inhibitors. The method they used for this was they took mouse bone marrow and they did a colony formation assay. And with PTPN11 G60R and E76K. And this was also in the presence of TNK2. So they treated the cells with a gradient of concentrations of dasatinib, and this was with or without the presence of GM-CSF, which JMML cells are hypersensitive to. The third test they did for this section, they investigated the role of TNK2 inhibition in colony assays. And they did this by transducing cells with mutated PTPN11 and wild-type or gatekeeper mutant TNK2. And this gatekeeper mutant was called T205I. So let's take a look at the results for this part. So the results for the first test were that there was increased colony formation when both were expressed compared to the mutant PTPN11 alone. So we can see that in figure 4A. So you can see that both mutants are the black bar, and you can see there's a lot more colony formation compared to all the other combinations. So basically both mutants, more colonies. For the second test, dasatinib reduced colony formation according to the dose. So we see this in figure 4B. So you can see that when there's no GM-CSF, obviously there's less colonies. But when you start introducing the drug, the therapy, dasatinib, the higher the concentration, the less colonies are able to form. And you see that go down with the gradient. For the third test, they found that TNK2 mutant increased, GM-CSF colony formation more than the wild-type TNK2 when co-expressed with the mutant PTPN11. And this is also seen in figure 4C. That is just used as a control. So we see that there's a lot of colonies because there's nothing being put there to inhibit it. And then we see the different concentrations of dasatinib used and how it affects the different combinations of mutants and wild types for PTPN11 and TNK2. So, however, they found that gatekeeper mutant TNK2 was less sensitive to being inhibited by dasatinib. So we can see that here. It's on we have the mutant version of the TNK2 on the second graph here and the fourth graph. So we can see that, dasatinib was just not as useful when you compare it to the wild-type TNK2 graphs. So there was also a reduction in the number of colonies formed in the presence of different TNK2 inhibitors and mutant PTPN11. And this suggested that PTPN11 relies on TNK2 for transformation potential. And we see this in figure 4D. So you can see that AIM100, XMD887, and dasatinib, these are all kinase inhibitors. Again, DMSO is just the control. So basically, we find that there are few inhibitors that can get TNK2. All right, so the next section is PTPN11 mutations confer dasatinib sensitivity in AML. So the first test we're going to talk about for this one is, while the focus has largely been on JMML so far, a significant number of AML cases exhibit mutant PTPN11. So this test aimed to test dasatinib sensitivity in AML patient samples. The method they used for this test is they compared mutant and wild-type PTPN11 patient samples, and they did ex vivo testing for dasatinib sensitivity by calculating the IC50 values, which we were talking about earlier. The second test they did was Sanger sequencing to reveal an AML-specific PTPN11 mutation called S502P. So this next test observed the transformation potential of this mutant for the AML disease. And they did this by transducing 293T17 cells, or mouse bone marrow cells, with the PTPN11 S502P, and they did immunoblotting to visualize this. So let's take a look at these results.